Good evening, everyone. I thought I would read you a bedtime story. Like a nightmare. Spy View by John Loftus, Blowing the Lid Off Lyme. How American Nazi spies have left a deadly, lasting imprint. The other night was our 41st wedding anniversary, and I was feeling well enough to skip the wheelchair and just go for a stroll with my wife, Susan. It turned out to be an eventful evening. Started out nicely. My home health aide dropped my wife and me off at our favorite seafood restaurant. The weather in St. Petersburg was so nice that we sat at an outdoor table. Our sneaky daughter had called ahead from New York and had cocktails and appetizers waiting. Life is tough, eh? After dinner, we strolled down the sidewalk past an old rundown hotel that had just been refurbished with several dazzling restaurants. I'd read a wonderful review about the new open-air patios, so we caught the elevator up to the rooftop. Beautiful view, nice tent-like cabanas all along the side. I liked the shade, so we headed there. I didn't notice the reserve sign. As we headed back towards the inside, a pair of women about our age waved us over and invited us to sit in their cabana. Lovely. Everything was going great. What could go wrong? The women were working with a private foundation to rehab rundown housing for the poor. They were new to St. Pete, so Susan said we would show them around. We handed them our cards. The younger woman looked at my card, and a strange look came over her face. You know, there's a famous John Loftus, she said. Susan smiled, her death's head grin, and noncommittally said, Oh, yeah? I know that look. I got up to move my walker or some such excuse. The woman said, No, really. There's a very famous guy with the same name. My wife smiled and gave in graciously. Yes, we know. That's him. I was trapped. The woman said, Really? You're the famous John Loftus? I said, I'm afraid I am. You have to understand, Susan and I don't get to go out much. And the last thing she wanted was to have our lovely evening interrupted by some fan gushing about how wonderful her husband is. I'm a nice guy, but I'm hardly a 2-cent celebrity. My wife saved lives as a psychotherapist, but no one comes rushing up to her in the street to tell her how wonderful she is. She puts up with me, no easy task. I was getting ready to make polite excuses and find another restaurant when the woman said, I'm from Cape Cod. I have Lyme disease, and my mother died of it. Susan waved at me to sit down. Later that night, Susan called our daughter Meg to describe the evening. And there was this lovely woman, and she wanted to thank your father for what he did for everyone who suffers from Lyme disease. Meg said, Dad? What did Dad have to do with Lyme disease? Sue replied that the woman said I was a hero to people suffering with Lyme. I was the first person to expose the connection to biological weapons research. Meg sounded a bit skeptical. How come I didn't know about this? When did all this happen? Susan said, You were very young. She was. It happened just after she was born. The perfect incubator. I quit the U.S. Department of Justice in disgust in 1981. I appeared on 60 Minutes in May of 1982, and my book, The Belarus Secret, came out a few months later. Something like 25 million people had seen my interview with Mike Wallace. After I blew the whistle on Nazis in America, it seemed everywhere I went, strangers would come up to talk to me. Some of them were spies. Apparently, I'd made a good impression with the American intelligence community. Instead of leaking secrets of state, I had done the right thing and fought with the CIA pre-publication censorship board for permission to release my book. Word had gotten out that I was one of the good guys. Now everyone wanted to tell me his secrets. This guy, he was different. A well-dressed businessman, serious. He said that I'd missed a few things. He told me how we'd accidentally dropped a nuclear warhead on U.S. soil and were lucky we didn't lose South Carolina. The other secret blunder was Lyme disease. After World War II, the Army had set up an artillery range at Fort Terry on Plum Island, New York, to test some of the captured Nazi biological weapons, including insect bombs filled with poisonous vermin. The island was a perfect testing ground, an ideal home for Plum Island Animal Disease Center. Nothing could escape, and most of the time, the prevailing winds blew everything out to sea anyways. The Army wanted to test just how far these insect vectors would go. It was supposed to be a harmless test. One of the ticks the Nazis found in North Africa caused an unusual rash that looked just like a bullseye. It didn't hurt anybody, that's what they thought anyways. It was just a way to measure how much damage an insect vector with real poison could inflict. But one of the Nazi scientists was a little careless, and the ticks got out at Plum Island. Somehow, maybe through a bird or a deer. Deer can swim a couple of miles in the ocean. The Nazi tick made it across the water from Plum Island and landed in the town of Lyme, Connecticut. A lot of people started getting the bullseye rash, but no one knew what it was. Then people started to get sick by the thousands. Then people started dying. The old spy, whose name I never knew, told me that the whole thing had been covered up. A gigantic study was done in New England under the guise of another disease, multiple sclerosis, I think he said. What they were really looking at was how many people were infected and how could Lyme disease be contained. The answer was it couldn't. The disease kept spreading and the army kept covering it up, hoping that it would not spread too fast or that someone would find a cure. Nazis in Plum Island. Now, this conversation must have taken place in 1982 because I included a short mention of it for the trade paperback edition of the Belarus Secret, which came out in 1982, early 1983. It was just a brief couple of paragraphs, but it caused a bit of controversy. On the leading Lyme disease, one of the leading Lyme disease experts, Michael Carroll, later recalled that in the book, The Belarus Secret, author John Loftus, the Justice Department employee who exposed Kurt Waldheim as a Nazi, states that Nazi germ warfare scientists had experimented with poison ticks dropped from planes to spread rare diseases. Loftus also states that he had received information that the United States had tested some of these poison ticks on Plum Island Artillery Range off the coast of Connecticut during the early part of the 1950s. As the controversy began to spread, a nurse who was one of the early Lyme victims clutched my book in rage as he demanded to know about the alleged Lyme cover-up at a national scientific conference. The spokesman from the U.S. Department of Agriculture dismissed the whole thing as some men from Mars conspiracy theories. The general response was that I seemed to be a rather sober and responsible attorney, but if I had fabricated this information about Lyme disease, then I should be prosecuted. Over the years, it's now become clear that someone should be prosecuted, all right, but it sure wasn't me. There's some evidence suggesting that Lyme disease did not even exist in America before the Plum Island experiments. Plum Island Lab Director Jerry Collis confirms that there was indeed secret tick research on Plum Island, but he claims that none of the poison ticks ever escaped. Quote, Plum Island experimented with ticks, but never outside of containment. We had a tick colony where you take them and feed them on the virus and breed ticks to see how many generations it would last and on until it's diluted. Recently, they reinstated the tick colony, unquote. It should be noted that the first bullseye rash in America was not reported until after World War II. Before then, there was not a single confirmed incident of Lyme disease, although there were some historical records that suggest a few people may have suffered similar illnesses. Certainly, the disease existed in Europe and Asia for centuries, but it was not prevalent in America, and certainly not widespread as it is now. The Texas tick in New England. And then there is the Texas tick that hitchhiked to Plum Island. Dr. Carroll wrote, quote, the focal point of experimentation on Plum Island in the 1970s, the Lone Star tick-like Lyme disease, is now spread throughout New York, New Jersey, and Connecticut. How did that happen? Unquote. A long overlooked document obtained from the files of an investigation by the office of former Long Island Congressman Thomas Downey sheds light on the second, more damning connection to Lyme disease. Congressman Downey had a lot of constituents dying of Lyme disease. Plum Island is off the coast of Long Island. He found a 1978 internal research document from the Department of Agriculture. Dr. Carroll explains, quote, in layman's terms, Plum Island was experimenting with the Lone Star tick, feeding them on viruses and testing them on pigs during the ground zero year of Lyme disease. They did not transmit African swine fever to pigs, said the document, but they might have transmitted BB Lyme disease to researchers or to the island's vectors, unquote. So the ticks did get out. They might have infected lab workers or even Plum Island's mice and waterfowl, the island's vectors, or possible disease carriers. Interestingly, the Texas tick they were playing with at Plum Island had never visited New England before. The Lone Star tick, named after the white star on the back of the female, is a hard tick, along with its cousin, the deer tick. It is a culprit in the spread of Lyme disease. Interestingly, at that time, the Lone Star tick's habitat was confined to Texas. Today, however, it's endemic throughout New York, Connecticut, and New Jersey, and no one can really explain how it migrated all the way from Texas. Dr. Carroll has an explanation. The Nazis did it, just as I'd written in my book back in 1982. Carroll explains, quote, a former employee at Plum Island in the 1950s has personal recollection of a Nazi scientist releasing ticks outdoors on Plum Island. A source who worked on Plum Island in the 1950s recalls that animal handlers and a scientist released ticks outdoors on the island. They called him the Nazi scientist when they came in 1951. They were inoculating these ticks, and a picture he once saw shows the animal handler pointing to the area on the Plum where they released the ticks, unquote. Dr. Erich Traub was one of the Nazi scientists brought to America in 1949 as part of Operation Paperclip. When I went public in 1982, no one even knew what caused Lyme disease. That same year, scientist Willy Berdorfer published an article in Science magazine for the first time linking a new bacterium as the causative agent of Lyme disease. The spirochete was later named Borrelia burgdorferi, or BB, in his honor. A spy's gift to America. I interviewed the stranger and started writing about Lyme disease in 1982, before the first article on ticks was ever published. It wasn't until 5 years later that scientists found the BB spirochetes in tick saliva, confirming that Lyme disease was indeed spread by tick bites. So why did I risk my reputation in 1982 writing about an unknown scandal at Plum Island? Why was I so sure the stranger I met was telling me the truth about the Nazi tick test that turned into Lyme disease? Well, for one thing, his story about dropping an A-bomb on South Carolina sure checked out. In 1958, a B-47 air crew on its way to England simply goofed up. The co-pilot went to get coffee while checking something, and instead of grabbing the ladder, he grabbed the bomb release mechanism. The plane just dropped it by accident on the little town of Mars Bluff, South Carolina. It didn't get much press attention at the time because the government said that the hydrogen bomb was empty. Its plutonium core had not even been loaded. That, my source said, was a lie. The conventional explosives detonated, leaving a 35-foot crater. 5 of the 7 nuclear fail-safe devices failed in the explosion. But the bomb did not go off. Only 2 little links were all that stood between a harmless accident and a nuclear vaporization of an entire American county. Yeah, my source knew what he was talking about. Like me, he had once held a Q clearance for nuclear weapons. There was another reason I trusted his information about the Nazi experiments on Plum Island. I had seen many similar reports in other secret archives back in the day. There's a top secret vault underneath the U.S. War College at Carlisle Barracks in Pennsylvania. Inside is a military study on how to plunge communist Russia into starvation by floating balloons filled with wheat rust virus over the grain fields of the Ukraine. There were other top secret reports on clandestine attacks on crops and animals, but the file folders are empty. Their contents have been destroyed without authorization or documentation, and I can guess why. The British had actually used the insect bombs tested at Plum Island. There is a record in the UN where the North Korean delegates screamed about overhead bombs exploding and scattering insects crawling all over the snow. Other records show that the Nazis may have tested similar bug bombs on the Russians. The Russian bug bombs were designed by the same guy who later conducted the tick experiments at Plum Island, Erich Traub. In fact, the Army even offered Traub command on Plum Island, but he turned the job down. After the war, the U.S. had agreed that America would get the Nazi rocket scientists, but the British would get the Nazi germ warfare experts. The British scientific liaison to the U.S. at the time of the Plum Island test was Donald MacLean, Kim Philby's associate in MI6 and fellow communist double agent. Traub was in Soviet hands after World War II, but somehow managed to escape into the custody of British intelligence. Communist spies, Philby and MacLean, then peddled Traub and, even worse, to Army intelligence. The reason was that America had to prepare defenses against bug bombs, and the British could not afford to pay for the research, but it seems they would share their Nazi scientists for some tests at Plum Island. Wasn't Kim Philby nice to us? Thank you, Nelson Rockefeller, for covering it all up. Let it be known, technically, Plum Island was under command of Nelson Rockefeller, President Eisenhower's Secretary of the Department of Agriculture. But the USDA always loaned the island for whatever the U.S. military wanted to test. Now, the funny thing is, Traub studied at Rockefeller's University before World War II. Later on, as Gerald Ford's vice president, Nelson Rockefeller headed up a special commission headed by the late Senator Ted Kennedy to try to block congressional investigation of intelligence abuses, which seems to have targeted Plum Island. One of the things that Senator Kennedy specifically wanted to investigate was biological weapon tests that may have harmed American health. Funny that Rockefeller owned a huge chicken breeding farm not far from Lyme, Connecticut. Funny that Traub published a paper on how chickens get infected with viruses. Now, these conclusions and the tips I got from my anonymous friend are not supported by everyone, including those who head national organizations dealing with Lyme disease research. Infections and syndromes similar to Lyme disease were found in early America, but scientists think that the deforestation killed off the carriers. Why did it suddenly appear again? Scientists aren't quite sure. Why were the first modern cases of Lyme disease concentrated on Long Island and in Connecticut, closest areas to Plum Island? Some people aren't quite sure. It's all probably just a coincidence, though. But my friend Rachel Vernon says you see hundreds of bunches of coincidences when you start researching Lyme disease. She will be furious when the British start opening up their files in 2022. Talk about coincidences. The British actually dropped bug bombs on Nazi Germany during World War II. Churchill himself authorized it, according to the secret files I read. I interviewed Germans who described how they were called out of school to pick potato bugs off their potato crop. My new friend, the lady whose mom died from Lyme disease, wants you to know that if you ever go to the state park at Truro on Cape Cod, that please stay in the marked trails. Don't go into the bushes. That's where the ticks are. The poison ticks are still there. There are signs warning people, she told me, but they're not very big. It's as if they don't want people to know. So, yeah, Patrick and I have been talking back and forth about phages, Lyme disease, and so I'm going to read an email exchange that between us. He actually sent this email out to the list. Sometimes I don't know if I'm tired or just brain dead because I know things. I know a shitload of things, but for some reason, the associations within Continuum never mesh the gears. Enter Teresa calling me all excited, reading me a scary bedtime story with highlights that she picked out specially for their significance. And I was reading to him from the Primer Shell game, which is, if I can find... the link. It's not that. Okay. Unscrambling the CDC's DNA patent profiteering bullshit. I will put this link down below. He says, it took a while and I made her reread it and we batted the ideas around like a severed head in a hockey rink, and slowly the image formed of what the problem is and how it was created and what we can do about it. For background, you have to have in the back of your mind the material from Loftus, which I just read, so that you have the history as a point of reference for what happened and what's going on today. From the link above, the Primer Shell game, it says in there that Lyme spirochetes did not evolve naturally and are genetically closest to an African bird borreliosis. From the Loftus article, it says the Army wanted to test how far these insect vectors would go. It was supposed to be a harmless test. One of the ticks the Nazis found in North Africa caused an unusual rash that looked just like a bullseye. It didn't hurt anybody. That's what they thought anyways. It's just a way to measure how much damage an insect vector with real poison could inflict. So for those who are real data dog diggers, we need a background on the parent spirochete to see what kinds of weaknesses we can exploit with the source material from which they derive their weapon. I know a few of you will appreciate that Babesia can easily be vectored by ticks, mosquitoes, and birds, as well as it being in the beef supply. So the bird connection with northern malaria was ever present, even if Borrelia was never an issue. That was the easy part. The hard part was when we were talking about the plasmids in Borrelia and the confusing array of nonsense that the article highlighted regarding testing. Sure, the article's a criminal indictment of fraud, but we need to get not to the only to the inferred implications of what they outline, but we actually need to know what is being technically referred to here, since it will make a difference on how we fight this beast. Bottom line, PCR tests are shit. They can be primed to look for something or whatever the fraudster chooses so that it will always show up positive. Or as the article goes on to say, that they can look for things that aren't there to say that it was negative when they merely look for things they knew were not there. The bottom line is the article indicates that if you test for the flagellin, the whip-tailed protein unique to these bastards, it will show up every time. So never look for that, and for good reason. I imagine that every man, woman, and fluid on the planet would light up like a Christmas tree full of flagellin protein. But that entire topic hovered on the situation over phage vectored plasmids. What is the difference between a vector and a plasmid? A plasmid is a small circular DNA that can replicate independently from chromosomal DNA within a cell. A vector is a more general term that we use to describe a vehicle that researchers use in the laboratory to introduce a DNA of interest into a host cell. A plasmid is a specific example of a vector. From the article linked above, phage vectored plasmids are variable DNA, like the outer surface protein A, not to be used for detection of human disease. The bad guys are trying to exchange how the taxonomy is performed and pretend that the phylum Spirocheta is ordered by its plasmids rather than the differences in flagellin. No one has ever seen such a thing before in science. Criminals are trying to change even the scientific history simply because they do not want to go to jail for a previous crime they committed, falsifying the case definition of the Burdorfer borreliosis and trying to put a fake vaccine on the market. So this is a part where we need more hand help. A bacteriophage is where they learned of plasmids that are infectious delivery system for the codes of the phages when they infect bacteria. It's claimed that bacteria have plasmids, but then that's like claiming that I have a wooden leg. I do, it just isn't attached to me. So are the plasmids integral parts of bacteria spirochetes, or are they insertions by phages? Never forget that you have helper, latent, lysogenic, and lactic phages. Could the plasmids in Borrelia be part of the organism or something from a phage, or something that the military put in there, since the homology has nothing whatsoever to do with the 90% of the life forms on this planet? What's so confusing about the excerpt above is that we don't know if we are looking for plasmids in Borrelia as part of a useless PCR test, or if they're looking for some kind of reaction in those automated test wells where the phage libraries are used to present proteins that are then tested for an industrial way, hundreds of variations at a time, to see what shows up. Or if that statement infers that the plasmids bound in Borrelia weren't native to the organism but were vectored there by phages, or if those phages were natural or DARPA weapons. We really need to get to the bottom of the nitpicking questions like this to get a handle on the problem, because as I was being rapid fire filled with facts by Teresa, even I was telling her to slow down. Something was nagging at me when she said, quote, plasmids change all the time, are bacteriophage vectored, and responsible for intra-kingdom gene transfer, unquote. That set the stage for what was to follow with our talk on the outer surface protein A. That is fungal antigen. That's always effing bothered me. It's exactly like the movies Alien, where a beast infects a host and takes on the genetic traits of the organism it infected. There should never be fungal antigens in a spirochete. Phylum, spirochete, kingdom, fungi. The damned alien is bursting out of our chest with acid for blood. But still, even if the DARPA MFers engineered that into the beast, the original plasmids would have to come from somewhere. That is where I took a break to put my cooked head ahead meals away, and it came to me: toxigenic phages. So if we synthesize the material up to this point in time, the Felix group that wanted $10,000 to fix you up with some enzyme supplements and hit you with a frequency to bring natural phages out of dormancy never addressed what was causing the little efforts to be dormant in the first place. He has someone on the list who was trying to get a hold of some bacteriophages from this company, the Felix group. I was ecstatic when I heard that the seeds of destruction in Borrelia and everything else for that matter were already there. We didn't have to reinvent the wheel or the axle, but something prevented it from expressing. We already knew that Borrelia had plasmids that didn't match any other life forms, that they were unnatural. But the one thing that I didn't consider is that knowledge of plasmids came from phages, and that toxigenic phages that I go on about all the time for things like tetanus, botulism, flesh-eating strep, diphtheria, pertussis, etc., are what makes these organisms deadly, because right now we are covered in the ephyrs but not dead. So the DARPA, I'm skipping over these words. I mean, and don't get me wrong, I do cuss like a sailor, but I'm trying not to. So DARPA had to have inserted or cultivated toxigenic phages in the Borrelia to make the fungal antigens. So this leaves us with a huge task of finding out how you can suppress the natural killer phages that would protect us from Borrelia, at the same time allowing the pet phages to make the organism toxigenic. It would be like a global government program to give everyone tetanus by shutting off the lactic phages of Clostridia, but still allowing the horseshit phages to make a lethal toxin to exist untouched so that they could do their damage. I can't figure out that bit. But if we could shut off the government toxigenic phages and turn on the endemic good phages, then there would be no Lyme. Tall order, and it took all of this time to see that relationship, but it is the only way that I can see how they did it and what we can do to stop it. Consider a world where everyone had tetanus and botulism, and then work backwards on what you might to correct it. All ideas are welcome. So he made me a little chart. Patrick made me this chart so I could try to present to show you the continuum. So we have here phages. We have Phriend and Phoe. On the Phriend side, we have lactic bacteria are forced to make copies of good phages that then kill bacteria when they lyse it to exit. Lysogenic or latent phage has been inserted by plasmids into genome of target but is not active for some reason. And the Phoe, toxigenic, bacteria lives, multiplies, puts out toxins, thus causing illness, tissue damage like flesh-eating bacteria, or death. Then we have Lyme disease. The spirochete contains phages that could lyse and destroy it, but for some reason the phage remains dormant, equals latent. Toxigenic phages may have inserted plasmids that code for the unusual substances like fungal toxins, outer surface protein A, and outer surface protein B. So Craig Venter seems to be able to switch genes within yeast on and off. Let's see if I can find that. Well, yes, here we go. I have so many links open, I apologize. Cyborg Yeast genes run by a computer. This was from 2011, BBC. Scientists have succeeded in forming a feedback loop between a computer and a common yeast to precisely control the switching on and off of specific genes. I will include this link down below. So a computer is a device made up of switches where the current is either on or off. The application of this technology then would be able to set up a living organism as a computing device, such as data processing, storage, and retrieval, but conversely could be used as a method of control of the entire organism to be puppeteered or merely as an on-off switch for the organism, such as punishment or death to anyone ultimately hooked to the Internet of Things. Phages to fight corona. I came across some different articles talking about using phages to fight the coronavirus. Phages predate bacteria, so the very notion of phages helping is preposterous on the face of it, but they never really explain their rationale. In 1930, transvection of plasmids by phages into a target host using restriction enzymes was discovered, and molecular biology was born. That became modern genetic engineering. With that as a sole background, then rethink how a virus that attacks a bacteria might have any therapeutic role against corona, a known genetically modified virus. They are genetic pathological liars with an agenda possibly revealed within our assembly of facts. Phages deliver plasmids that are circular DNA capable of either directing operations from the cytoplasm or becoming inserted into the host DNA, similar to what is called an endogenous retrovirus. This is the model for the use of viruses as vectors for gene therapy, of which all of the trials ended in the test subject's death. So there is a theory that what the controllers are attempting is to rewrite the human genome, either via double-stranded DNA edits, such as CRISPR, that itself was discovered as an antiviral defense mechanism of bacteria by DuPont and their yogurt division of their military weapons corporation, or the insertion of a third strand DNA. Yes, I was looking. Hold on. Yeah, it's CRISPR, splicing and dicing the genome engineering in the CRISPR revolution. Oh, but don't worry, it won't change your DNA. Yeah. Failures at either modification technique would answer why all attempts at coronavirus vaccines have failed. Could it be that the messenger RNA abomination called a vaccine when it's not is the response to failed earlier attempts to splice their corruption into the human genome, thus killing the rest of the test subjects with the plausible deniability of blaming it on the coronavirus? Could it be that all coronavirus research, which is based on a mild cold virus that was militarily enhanced with gain-of-function techniques, might have been part of a binary or even tertiary weapons platform? Cross-platforms applications. It's always helpful to organize data chronologically to see the stepwise development of a process or an idea. Rabies is an insignificant but well-known virus that afflicts mammals, including bats. Pasteur was the first insane madman to achieve gain of function with his rabies that he had to force into rabbit brains. Once he accomplished his goal, he was able to make a strain that consistently could incubate within a week. For comparison, there's a record of a man who did not show symptoms of rabies for 30 years after he was bitten. Gain of function is just a fancy term for weaponizing biological organisms. Pasteur was certainly not the first to use bioweapons, but he set the stage for gain of function, and he did so with rabies, which we will follow like a golden thread. Viruses are susceptible to vitamin C. This includes smallpox, rabies, and others. Humans and bats are unique in that they do not produce vitamin C within their own bodies. It makes a perfect choice to use bats as a platform for weapons development. We see this in the admission that the Wuhan coronavirus was claimed to have arisen in a bat, and that the level 4 biohazard facility was doing gain of function on your tax money given to them by Fauci. Also of note is that the ACE2 receptor, that just happened to be the magnet of choice for targeting human cells, also appears in brain tissue. the brain being the end target of a nice foamy rabies infection. Seeing any patterns yet? Corona also codes for HIV, which in Patrick Jordan's theory of continuum gets us chasing at high speed around the circle of facts where the rabies virus, being a large vessel, is able to be husked out so that new genetic material can be duct taped in. They have been doing this with smallpox, HIV, and malaria with the excuse that they are trying to create delivery vehicles for vaccines. But where have we heard the notion of using viruses as delivery vehicles? Bacteriophages as plasmid vectors, adenovirus as gene therapy vectors, now rabies as a way to sneak in chimeric rewrites. The U.S. military in their African operations tested a malaria vaccine using rabies vaccine as a control. A control, by definition, means that you give nothing to the other group. The study revealed that chloroquine interfered with the production of antibodies against the rabies vaccine. Some facts are just too bold to ignore. Chloroquine, like the hydroxychloroquine that in the beginning, the Trump even said cured the coronavirus, but no, we couldn't have that because we had some therapeutics and some vaccines that we had to roll out. I digress. In 2005, Thomas Jefferson University was doing illegal gain-of-function experiments on rabies because they wanted an already lethal abomination to be able to penetrate the blood-brain barrier better, the BBBB. Their claim, as all of their claims, was that they were doing it for health. If they could create a chimera on the inside of a rabies delivery vehicle that looks at the molecular level like a bullet, then they can insert whatever their payload was into the brain with no resistance. With a tweak of a single amino acid, they were able to do that. The world is not any safer, and exotic diseases are now rampant. Let me go over here and show you this link. Now, these PDFs, if you are interested, send Patrick or I an email, and we could send you the links to this. Okay, hold on. It doesn't want to open for me. There we go. Single amino acid change in rabies virus glycoprotein increases virus spread and enhances virus pathogenicity. The results are consistent with the notion that the rabies virus glycoprotein is a major contributor to the rabies virus pathogenesis, and the more pathogenic RVs escape the host responses by a faster spread than that of less pathogenic rabies virus. So, yes, there you have it. So, let me go on back down here. But why rabies? Why gain of function? Why attempting to go where no man has gone before? Because the best way to defeat humanity is to gain access to the central nervous system to either rewrite the genetic code in the brain or to install cybernetic machinery in places that, for a good reason and many millennia, have been walled off to the outside world of demons. Not to worry, though, transgenic mice with humanized brain tissue have been developed, and that is how we discovered that there are ACE receptors in nervous tissue, and the only reason we went looking is that current chatter in the media is that there are now neurological complications to the so-called corona infection. I like to call it the corona genetic rewrite. We also found that MERS, the beta test for the corona weapon uses different binding site. But this would be typical of revision of software to get rid of bugs and increase efficiency of the program. Things to ponder. Both rabies and corona have been subjected to gain of function experiments. And both rabies and corona cause central nervous system disease. Both rabies and corona may be listed on a label of clear fluid, but if you didn't make it yourself, you have no idea what's inside that vial, let alone what might be inside the rabies virus That could be any genetic material or the corona that was modified with HIV. Messenger RNA is not a vaccine, it's a genetic rewriting tool. Even if the label on the cleared fluid says messenger RNA, unless you made it yourself, you have no idea what is in it. Clearly, everything surrounding this material is an act of war. There is no law during war, only necessity. Do what is necessary. Yeah, we are attempting to do that. I just wanted to point out too, here we have this renowned tick immunobiologist to develop Lyme disease vaccine with a federal grant. Dr. Powell wants to adapt the rabies vaccination platform to produce antibodies that can protect against Borrelia burgdorferi, the bacteria responsible for Lyme disease. The intention is to apply this work to other tick-borne diseases in the future. Yes. We are using the rabies virus as a delivery platform to send in some vaccine candidates. Absolutely, completely insane. Viral vaccines are produced in 3 ways. Live attenuated, which is rendered incapable of producing full-blown infection. Inactivated virus, which is more popular because of its safety and efficacy, and is the platform for the actual rabies vaccine. And the virus-like particle, which is created by extracting a protein from the virus and producing a shell with viral proteins on the outside, tricking the body into developing defenses against the virus itself. Yeah. So, I'd love to hear what you guys think. If you would like copies of these PDFs, I will include my email down below, as well as Patrick's. Let me know what you guys think. I appreciate you taking the time to listen.